So, good morning, everyone. It's my pleasure to just get into this lecture in the last part of the PMS 300 reproduction part. So, just to introduce myself first, my name is L53. As you can see here, my email address is indicated. I'm a faculty member at the Animal Science, Animal Reproduction in Biotechnology Lab, and my office is at Focus campus. So this is my first lecture at all in CSU. So just to give you some background information about myself, just to know who is standing in front of you. Just I just did my bachelor's originally in Ethiopia, and did my bachelor's at the University of Alamaya in Ethiopia, and then I just moved to Germany to do my master's and PhDs in animal reproduction biotechnology. And then I was there the last 15 years as a faculty member, and then just moved to CSU the last October 2019. So I'm almost 1.5 years here at CSU. So my main area of research interest is kind of embryology. That means I am interested why some embryos are surviving, why the others are dying. This because of the fact that the embryo mortality is the main factor which affects fertility in dairy cows, the model which I use for my study, and also mainly also in the human reproduction. So just to give you that one as a background, and then whenever you have any questions, you can chat during the lecture to the assistant Avi, and then she can forward it to me, or she can answer to you. But in the meantime, you have also the chance to send me an email whenever you need any kind of explanation on my lectures. For today, so I have already done the introduction about myself. So I will try to give you some comments or some concepts about reproduction as a general, and then I will go on the different sections of reproduction, starting from gametogenesis, how gametogenesis, or the production of gametes, takes place in the testes and also in the ovaries, and once the embryo exists, what kind of sex determination is happening, at which level the sex determination is happening, that is at chromosomal level sex determination, or gonadal or primary sex determination, or the last one is the phenotypic or secondary sex determination. And then afterwards, we'll just look into the main regulatory mechanisms in the hypothalamus pituitary gonadal axis, both in male and female reproduction, and later on, finally, today's will be the various types of abnormalities associated with sexual differentiation in human. Just to give you some ideas what we mean by reproduction. So we have reproduction here, and the central point in reproduction is the unification of the gametes, that means the sperm, and in one side the egg in the other side. So when they are unified, that means at the point of fertilization, the new individual or combination of genes will happen. That means the zygote. So this zygote, which is a one-cell structure, can grow through various types of processes, cleavage and processes, multiplication of cells, to the blastocyst first, and then transferred to implant, and then comes an offspring, an individual. So the very important, or the very exciting and interesting part of this whole process is that this one cell zygote, which is a result of the unification of the sperm cells, the egg, give rise to an individual with multitude of cells at adult level, there are around 200 different types of cells in the individual. So the process, which looks to be simple, is very complex and very interesting. So where does it start from the sperm? The sperm is coming from the gonad called testis, and the egg is coming from the gonad called ovary. So these are the gonads, which are the organs where gametogenesis happens in both cases. So gametogenesis is the process in which sperm in the testis, the egg or the ova in the ovary are produced to be ready for fertilization and produce the zygote. So once the zygote is formed, then the next step here is that how the sex of the zygotes will be determined. That means sex determination. Yeah, so we will have this part in our talk today, and we will have this part in our talk today. So that means sex determination can happen at chromosomal level, depending on the chromosomal setup, or at gonadal level. That means the chromosomal arrangement or the chromosomal combination determines which type of gonad, either an ovary or a testis, will develop in the gonads, as a gonad. And the other one is when the gonads reach to its ability to produce steroids or hormones, then these give rise to the phenotypic sex determination. So in our case, today, we will be trying to see how gametogenesis is happening in the ovary and the testis. Where are the differences? Where are the similarities to generate a sperm which is able to fertilize the egg, which is a round-like structure. So this will give rise to the zygote. So this shows that reproduction seems to be a complex and a very interesting aspect, which we are going to see in the next lecture hours. So what I am planning to do in these lectures is the first 5 lectures, I will try to cover most of the items which are important for you, to just to give you an idea how different aspects of reproduction looks like. And then the 6th day of the lecture, I will try to review what you have learned or what I have taught in the first 5 lectures, the very important ones. And then later on, the last part, I will try to discuss with you guys kind of preparation for the exam in collaboration with Abby. We will do here in the last part of the lecture next Friday. I mean, next week, Friday. So as I said before, so the normal reproduction process is very important for giving rise to healthy offspring, which is also very important for the survival of the species, because reproduction gives genetics to the new offspring, so that the genetic will be able to survive in different environments, especially in changing environments. So reproduction is starting with the merging of parental chromosomes, which provide biological variations in individuals that is necessary for species adaptation and survival under changing environment. So as I said before, the process of reproduction includes the production of the male and the female gametes, how they are processed, how they develop, how they grow, and how they unite to form the new entity of individual. So as I said before, the gametes are produced in the gonads with the process of gametogenesis, either in the ovary to give rise to a competent oocyte, or in the testis to give rise to a sperm which is able to fertilize the egg. So during gametogenesis, the first step of gametogenesis is the generation of the primary oocytes, spermatocytes in the testis. That we are talking about now, gametogenesis in the testis, which is start with mitosis. Mitosis is simply multiplication of the primordial germ cells using the process of mitosis, giving rise to daughter cells which are completely similar to the original ones. So the mitosis of the primordial germ cells, and then the primordial germ cells are generating the primary spermatocyte, which have 46 chromosomes as a diploid. So the first meiotic division happens right after that, and during the first meiotic division, the maternal and the paternal chromosomes here, the homologous chromosomes, are arranging themselves around the equator of the cell so that the next step of meiotic division starts. So this arrangement, or the closure of the two of the homologous chromosomes, is paving the way for a certain kind of process to happen. That is the so-called crossing over. Crossing over is a process in which homologous chromosomal recombination happens. That means non-sister chromatids. These are the two sister chromatids of one of the homologous chromosomes, and the non-sister chromatids are exchanging segments of the chromosome so that the sister chromatids are no more identical to each other. So this crossing over is one of the very critical event in the meiotic one, which leads to genetic diversity. So this chromosomal combination after crossing over is no more similar with the previous one. So once the crossing over happens, there will be separation of the two into two. I mean, after crossing over, the first meiotic division will be complete. The first meiotic division, when it is complete, we will have secondary spermatocytes, and as two secondary spermatocytes will be generated. That means when the first meiotic division is complete, we have secondary spermatocytes. The secondary spermatocyte, each of them contains 23 chromosomes, each with two sister chromatids. Following that, the second meiotic division happens following that, and this will lead to the separation of this secondary spermatocyte into spermatids. The spermatids contain 23, or half of the chromosomes of the diploid, so that they will have only half of the chromosomes, which will be considered later on as a genetic material from the paternal side. So looking into So the very important aspect here is that from a single primary spermatocyte, we have four spermatids which differentiate into 4 sperm cells. We need to consider that this is only one primary spermatocyte. There are millions of spermatocytes in the gonads which are producing millions of sperms at a time. So the very important summary in this one is that, so the gametogenesis in the testis starts with mitosis, which give rise to primary spermatocyte, and with the meiosis 1, this will give rise to the secondary spermatocyte, and the other one is the secondary spermatocytes are divided again in meiosis 2 to give rise to spermatids. And these spermatids are differentiating to give rise to sperm. So this sperm is a haploid. That means that it carries only half of the chromosome of the human. That means 23 chromosomes compared to the previous or the original part. So this is how gametogenesis or spermatogenesis happens in the testis. So when we look into the gametogenesis in the ovary, in the female, then we see the principles are the same. That means the mitosis of So the mitosis of the primordial cells give rise to the primary oocytes. And these primary oocytes again go to the first meiotic division, which is also followed by crossing over, like in the sperms, what we have seen before. And the secondary, the first meiotic division of the primary oocytes give rise to secondary oocytes. These are oocytes which are normally in the ovary ovulating. That means these are matured oocytes which have complete their first meiotic process. And the second meiotic process, divisions, happens in the female or in the ovary after the oocyte is released into the oviduct. That is the fallopian tube. We will see about that when we are talking about the female reproduction. And in the oviduct, the secondary meiotic division happens in the presence of sperm. Then that gives rise to the secondary oocytes, which have its both polar bodies are here. So we have in the first meiotic division, the secondary oocytes will take the majority of the cytoplasm, and then some portion of the cell will take the very tiny portion of the cytoplasm, but the half of the genetics of that specific cell, and that is called the first polar body. This first polar body is functionally, they are not relevant. They will degenerate. But the secondary oocyte goes to the second meiotic division after fertilization. Once the second meiotic division happens here, so the sperm cells or the sperm nucleus is going in here, and the second meiotic division happens, and then give rise to the combination of the sperm nucleus with the haploid of that specific secondary oocyte, and the second polar body, which is haploid of the secondary oocytes, will be released into the place. So these secondary oocytes, again, secondary polar body, like the first polar body, are both of them degenerate and they are functionally not relevant. So just to give you a comparative presentation of the gametogenesis in males versus females, we see here when these processes are happening. As you can see here, the mitosis of the germ cells happens in the male, in the embryo part, a little bit in the embryo part, but the majority is happening when the individual is at puberty. That means in sexually adult reproductive system. However, in the female, so the mitosis and part of the meiosis, which give rise to the primary oocytes, happen during the embryonic phase. That means during the fetal period. So baby girls are mostly born with the primary oocytes. So when they reach to the puberty or sexual maturity, this process will continue, and the first meiotic division happens, completed, and the secondary oocytes will be there so that there will be ovulation when that specific girl is at the age of her reproductive age. Again, the first polar body will be released here. The egg will be released into from the ovary through ovulation, and in the oviduct, the fertilization happens from the using the sperm, which is already released from the testis, and the newly formed zygote will be formed. So here is very important to see that a single spermatocyte or spermatogonial or spermatocyte, primary spermatocyte, give rise to 4 different types of sperm cells. While in the female, the primary oocytes, one primary oocyte give rise only to 1 secondary oocyte, and finally 1 which is ready for fertilization. So these are the 2 basic differences between gametogenesis in the male and in the female. So just to give you, before that, I just forgot to show here one of the other variabilities which happen. I said before, one of the genetic variabilities happen during the process of meiosis is because of the crossing over between the non-sister chromatids. The other variability is the random distribution of maternal and paternal chromosomes into 2 of the secondary spermatocytes, or in the case of oocytes, also in the secondary oocytes. So these 2 events, which are important for the sources of genetic variability between individuals coming from the same mother and father, is the 2 events which are happening in the first meiotic division. The first one is crossing over, which is a crossing over or the exchange of segments of the chromosomes of non-sister chromatids. And the other one is the random distribution of maternal and paternal chromatid pairs between the 2 daughter cells which are divided during the meiosis period. So this is very important aspect which one has to keep in mind, that how a certain genetic variability arises during the process of gametogenesis. Crossing over, random distribution of the maternal and paternal chromatids in pairs between the 2 daughter cells. So one of the exciting part in reproduction is that once gametogenesis is complete and the sperm is coming from the testis to the oviduct, and the oocyte is matured in the ovary and released into the oviduct through the process of ovulation, and the unification of both the sperm and the egg in the oviduct is the beginning of the pregnancy. So the lady or the woman is said to be pregnant starting from that specific day, where you have a unification of the sperm and the egg in the oviduct. One of the exciting things, that means the sex of the embryo at a time will be fixed in the time of fertilization. That means, so whether that specific entity or that specific new life, whether it is a male or a female, depends on the combination of chromosomes at the time of fertilization. But it's always exciting to see that what is the sex of a specific boy or girl. Is she a boy or girl is always the question which is asked. So this is very important because of the fact that the sex of an individual, whether it is male or female, it determines whether who we are and how we live. So it's very important. So looking into that, sex determination can be determined at different levels. The very important one is that sexual differentiation or sex determination is important because of the fact that the development of male or female reproductive organs or whatsoever characteristics or behaviors are determined whether that specific embryo is having a chromosomal combination which give rise to a male or which give rise to the female. So the first is the chromosomal sex determination, is also called genetic sex determination. So once the genetic sex determination is fixed, then that will give rise to the development of a gonadal sex determination. Gonadal sex determination is that whether a specific, whether the tissue is developing to the ovary or to a testis. So once the gonads are differentiated, once we know that the sex of the gonad is either ovary or a testis, then when these gonads are attaining their endocrine function, they give rise to the various types of phenotypical sex determination. This phenotypical sex determination that is called the secondary, that is a primary sex differentiation. The other one is the secondary sex differentiation. That is mostly the development of internal reproductive tracts that can be either the different components of the male, that like epididymis, or the different parts of the male reproductive organ, or it can be also differentiated into oviduct or uterus, which are all important for as an internal reproductive tract. And the internal reproductive tract and also external genitalia are also the ones which are resulted from the differentiation of the gonads, either in the direction of ovary or in the direction of the testis. So the phenotypic sex differentiation is either internal genitalia, internal reproductive organs, or external genitalia. So when we look into the chromosomal sex determination, so we see that this is the karyotype of the human chromosomes. The human chromosome contains hope that everybody is following me. Am I clear? Okay. So the chromosomal sex determination is starts at the chromosomal level. So the karyotype of the human chromosomes indicate that the human chromosomes, human cells contain 22 autosomes. These are chromosomes which are not sex chromosomes. Normally, sex chromosome is a confusing term because most of the genes which are, so the other ones are the two sex chromosomes, either X or Y in case of human. So as you can see here, the different types of chromosomes which are karyotyped in human are different in their length. The shortest one is the Y chromosome, which is here, and compared to the X chromosome, which is homologous to it, it is relatively large. So it has a lot of genes on it. So the sex chromosomes, is the name by itself is relatively biased because that sex determination is not only determined, so because most of the genes which are important for sexual development are located on these autosomal chromosomes. That's why sex chromosomes are, since the term by itself is relatively confusing, but it is agreed to be used like that. So we have 22 autosomes and 2 sex chromosomes, which are X and Y. So that means that so sex chromosomes can be inherited from the parents, so that the individual will have either a male or a female chromosomal combinations. So the female parents, which are designated as XX, they have both chromosomes, X chromosomes. So the gametes which are produced by these female parents, they have both X on their chromosomes. However, the male parents are producing either sperm cells carrying the X chromosome or the Y chromosomes. So offsprings which are inheriting X chromosomes from both are becoming female, and those offsprings which are coming from these parents taking a Y chromosome from the father and the X chromosomes from the mother, they are male offsprings. That means that XX individuals are female and the XY individuals are mostly genotypically males. So this chromosomal determination or chromosomal sex determination is varying between different types of species. So for example, the XX and XY is mostly used in mammals. This is an example for human, that the male gametes are either X or Y, the female gametes are XX. So there are different combinations of offsprings. So offsprings carrying XX are the females. Offsprings carrying XY are the females, the males. So the other chromosomal combination in insects is, which is very evident in insects, is XXXO. Sorry. So the other one is, in insects, is a totally different story. That means they have XX or XO. That means the absence of one of the chromosomes leads to the maleness. When both chromosomes are available, then they will be females. So here, that In insects, when the two gametes produced have XX, but in the male gametes, either they have one X chromosome or they have no chromosomes. The combination of that leads to such kind of combinations. The other combination is that in birds, for example, they have a different types of, an opposite type of chromosomal combination like the humans. In case of birds, the combination is like the ones, for example, which we have seen in human. XY gives rise to the male, but in this case, the two asymmetric types of chromosomes are giving rise to the female. So this is just an example to see that how chromosomal sex determination is varying between different types of species and animal kingdoms. But for us, this one is interesting and important, which we go on doing. So, as we said, the first part of this was the one which we said chromosomal sex determination, which I showed here. So the first part, what we have seen, was the chromosomal sex determination. This depends on the combination of X and Y chromosomes. If an individual is XX, it will be female. If an individual is XY, it is a male. So how this chromosomal combination or genetic combination is affecting the second phase or the primary sex determination, that is the second one is the gonadal sex determination. As you can see here, the differentiation of the gonad either to the ovary or to the testis starts from a kind of gonad which is called bipotential. That means it has a potential to be either developed to the direction of the ovary or it can develop to the direction of the testis. This depends on the activity of the genes, which is also governed by the combination of chromosomes. In the presence of Y chromosomes, that means in XY, this is a SRY gene. It is a gene which is located on the Y chromosome. It is called SRY gene. It's a called sex determining region in Y chromosomes. So this SRY gene will be coded from the Y chromosome of the individual, which is it's also called a testis determining factor. This is a transcription factor which is activating the SOX9 gene in the gonad, and the SOX9 together with the SR1 gene are pushing the development of the testis. How do they do that? Normally, the other part is that in XX combinations, we have the R-spondin gene, which is very important transcription factor, activates the Wnt4 and the beta-catenin pathway, which is important for the development of the ovary. When the XY combination or when the SRY gene is active, there will be the SOX9 gene, which inhibits this pathway so that only the testis will be developed in the undifferentiated gonads. In the absence of this one, when this one is not there, the default program in the gonadal development is this pathway, which is mainly governed, or which is mainly executed by the R-spondin gene 1, which is located on chromosome 1 of the human genome, regulates the activity of the Wnt and beta-catenin signaling, which are very important for the development of the ovary. So we here use So from the undifferentiated bipotential gonad, we have finally either a differentiated gonad with ovary on this part, and this one is the mesonephrons. This is a place of the different types of ducts which are important for the development of the internal reproductive organs, either in the direction of the male or in the direction of the female. So as I said, this is a gonad which is influenced by the SRY gene, and then also SOX9 give rise to the testis which is developing here. So in the undifferentiated gonads, we see here how gonadal differentiation is directed by the presence of an SRY gene. SRY gene, which is giving rise to, or which is coding for SRY protein, which is also called a testis determining factor, is acting on the undifferentiated gonad. The undifferentiated gonad contains several types of, so before, can I just go back once more, just to see. The bipotential undifferentiated gonad contains different types of cells in its content. One is the three types of cells are, this is the bipotential gonad, contains as a cell, for example, the somatic support cells. It has also precursor steroidogenic cells. So the third ones are primordial cells. These are the ones which are becoming supporting cells. The precursor steroidogenic cells are the ones which are becoming components of either the testis or the ovary to produce different types of steroids in the ovary or the testis. The primordial germ cells are the ones which become sperm or an ovum. So when it comes to in the female, in the presence of R-spondin, one on which activates the Wnt4 and the beta-catenin. The somatic support cells are differentiated to granulosa cells. And the precursor steroidogenic cells are differentiating to TECA cells, and the primordial germ cells are differentiating to ogonia, or the precursor oocytes. So in case of female, in males, combination with the presence of SRY gene, the development of these different types of cells takes a different direction. This will be the somatic supporting cells will be Sertoli cells, and the other ones will be the precursor steroidogenic cells are the Leydig cells, and the other ones will become spermatogonium. So the different types of cells which are available in the bipotential gonad are developing to different directions depending on the chromosomal combinations of an individual or the chromosomal sex determination which we see here. When we look into the direction which is followed by in the male, when the SRY gene is activated, there will be a coding for the SR1 protein, which is a testis determining factor, and acts on the undifferentiated gonads. The undifferentiated gonad have the potential spermatogonial cells in the middle and the potential ogonial cells in the periphery of the undifferentiated gonads. So in the presence of the SRY protein, the Leydig cells are activated to produce testosterone, and this testosterone will be used for continuation of the development of the different male somatic organs or development of mental development in the direction of a man. The other ones are the Sertoli cells, which are found in between the different spermatogonial cells, are producing, with the action of the SRY gene, they are producing anti-Mullerian duct factor that is called anti-Mullerian hormone, which is reducing the potential of development of the Mullerian duct into the oviduct. So this is just to show you an example how the presence of SR1 gene affects the somatic cells to be steroidogenic cells and produce steroids, which are important for further development of the secondary phenotypic characteristics. So the phenotypic sex of an individual is again dependent on the gonadal sex, whether it is the ovary, which is there, active, or is it the testis. So the mesonephros, which is the undifferentiated part, is the one which is containing the different types of ducts. The very important two ducts are the Mullerian duct and the Wolffian duct. And the genital ridge is the one which is becoming either ovary or a testis. So when there is a male direction of development, so in the males, there are two hormones which are generated from the which are developing from the two types of somatic cells. One is the testosterone, which is produced by the Leydig cells. And the other one is anti-Mullerian hormone, which is produced by the Sertoli cells. There is a clear distinction of job or distribution of job between the two cells to support the development of the male reproductive organs. So the presence of testosterone will activate or will support the Wolffian duct, which is red, which is blue here, to develop into the male direction. That means it will develop to epididymis, it will develop to the vas deferens or the seminal vesicles. So in the presence of testosterone, the Wolffian duct will develop into these three components of the male internal reproductive organs. The anti-Mullerian hormone is playing a role in inducing degeneration of the Mullerian duct, as its name also indicates that it is so anti-Mullerian hormone is involved in degeneration of the Mullerian duct, which is normally give rise to the development of the female side of the internal reproductive organs. when these two hormones are not there, when these two hormones are absent in case of female, because of the XX chromosomal combination, so the male, the females, the Wolffian duct will regress because of the absence of testosterone. The Mullerian duct will not regress because of the absence of anti-Mullerian hormone. That's why the Mullerian duct will develop into a female internal reproductive organ, which is mostly composed of the oviduct, the uterus, and some portion of the vagina. So this is how the Mullerian and the different types of ducts are developing to a different internal organs depending on the presence of either ovary or a testis in the genital ridge, which is also dependent on the chromosomal sex determination part of the process. Do we have time, or is that?
Yeah, 1 minute over.
1 minute over, okay. So I think that is all for today. Maybe next time we will go on continuing that one, the sex determination, especially the phenotypic sex determination, and we will talk about the gonadal and pituitary, the hypothalamus pituitary gonadal axis, how that regulates the different types of reproduction in males and females. And also we'll look into the different combinations or abnormalities which happen in the process of gametogenesis or during fertilization, how these different types of sexual differentiation abnormalities looks like, just to show you that. That's all for today. I think if there is any question, Abby will be available on the chat. And thank you all. If there is any need, as I said before, you can email me, or you can email the graduate assistants here. They will handle all the questions. Thank you very much, and see you tomorrow.